So now that we left off last with transcription, we have to pause to take a look at what we have now that we're done with transcription. Remember that we have left our DNA inside of the nucleus, so what we have is a copy of instructions that are alternate complementary opposites of what was in the DNA. Now remember, we were not copying the entire DNA strand, just one gene. So to put that in perspective, we're stuck here. Those are our original instructions with our DNA. We have made a copy of that, the mRNA. This is a messenger here, our messenger RNA. It's going to tell our cell what to do, but that must leave the nucleus and go out into our cytoplasm to where we need to actually do what it says. Now remember, these genes we've said are instructions for eye color, for hair color, for making certain enzymes, for pigmentation, etc. But in reality, what we're going to see is that they really are instructions just for building simply polypeptides, which are really proteins. So let's take a look at how we would do that. Now remember, this was a two-part play. In our first part of the play, transcription, to scribe or to make a script, we were just copying the directions onto mRNA. Now we've got to translate that, which think about what translate means in your foreign language classes. Take a message or statements that are in one language and translate them into a totally different language. And that's really what our cell is going to be doing. We're going to take the instructions in mRNA and do what they say, translate them into actual proteins using something called tRNA. Oh my gosh, there's another one and that's confusing, but note, this isn't mRNA, it's T for transfer. It's going to have a job, and I don't want to scare you, but I want to show you a picture of what it looks like in action. It looks like a train wreck, but bear with me. Whoa. What is that? Well, we know what a couple things are. This is out in our cytoplasm. There's our mRNA. This brown guy is our ribosome, which ribosomes, we said, is their job to build proteins. All this other stuff, oh my gosh, what the heck is this? We'll get there and we'll try to understand that. But for now, this is a snapshot of our cells in the process of building a polypeptide chain, or a protein. Because remember, a protein is just made up of hundreds and hundreds of amino acids stuck together. The proteins that make up your muscles, muscle fibers, made of amino acids. The pigmentation that gives you hair color and eye color, made of amino acids. The enzymes that do chemical reactions in your body, made of amino acids. You get the idea here. So let's zoom in on this and focus on exactly how this has happened, because this looks kind of messy. So before we can do any of this, we've got to look at the two terms here that are key, codons and anticodons. Now if we look at the term codon, it looks almost as if it actually has the word code in it. So there's something here that's a code, and anti means opposite code. So if we think of those mRNA molecules, remember they're made up of bases, A's, C's, G's, and U's, that holds a code. So in them, if we read them 3 bases at a time, each one of those is called a codon because it's a secret code for something. So again, there's our mRNA molecule, only one strand. If we read 3 bases at a time, that's a codon. So GCU is a codon that codes for something. ACG is a codon that codes for something. So in essence, the secret code is telling our cell to do something. And we're going to look at what that actually codes for in a moment. But with those codons, it's if we think of it, we're reading 3 letters at a time. It's just like reading the words in a sentence. If you look at this sentence here, which is stupid and really means nothing, if I read this 3 bases at a time, 3 letters at a time, the fat cat ate the red rat, it makes sense. It's telling you that a little story, if you will, about this fat, obese feline that devours a obviously strange red-colored rat. But really, our codons and our mRNA is going to work the same way. We read them 3 bases at a time, and that's going to hold a code for instructions. So really what that code means, and I'll always give you one of these charts, is that each one of those codons, if you think about it, codes for directions to build a protein. So what are our proteins made up of? Amino acids. So really each codon codes for a particular amino acid. Now this might look a little bit confusing here, but what we want to see is that all we've got to do is look at particular codons to see what they code for and look them up in this chart that I'll always give you. So let's say one of our codons are G, C, A. How am I supposed to know what that codes for? Well, our first amino acid, our first letter is G, so we know we're going to focus in this area. It's going to code for an amino acid that's somewhere over in here. Our second letter, we go to the outer second ring, and here is a C. So we know we're focusing right here. G, C. And our last base, we go out to the third rung, is an A. So G, C, A codes for the amino acid. Its instructions, it's calling for alanine, one particular amino acid. So we can see it's actually pretty simple. Each of these three letters, in essence, simply codes for a particular specific amino acid. Now, how do those amino acids get there? That's where this other molecule of RNA, tRNA, or transfer RNA, comes from. Its job is to transfer or transport amino acids. And what we're going to see is that the unique structure, here is our weird clover-shaped tRNA. Notice it's made up of U's, A's, C's, and G's. It is carrying an amino acid at the top. This one happens to be carrying one called phenylalanine. Another one could be carrying our friend alanine. One could be carrying leucine, valine, you name it, any of the 20 amino acids. But how does this know where to go? And the secret is at the bottom right here. Our anti-codon, our opposite codon, AAA. Because what's going to happen here is that this AAA, do you remember what bases are opposite and complementary to AAA? If you remember, U's are complementary to adenine. So the codon that this is going to be attracted to would be uracil, uracil, uracil. Let's take a look at how that actually works. So during this process of translation, what we're going to see is these tRNA molecules come in and match up with codons on the mRNA. The tRNA's anti-opposite codons match up with the codons in mRNA. And it happens in the ribosome, because these are going to be our builders. These guys are our construction workers that are going to read the directions and actually build the proteins. So what we can see, if we look at this a little bit more closely, is very simply, here is our anti-codon at the bottom of a tRNA molecule. There's our codon. So if you actually go and look up in the wheel, AUG codes for the amino acid met, or methionine. These three letters right here are actually coding for methionine. So all that happens, here's our tRNA molecule. It brings in methionine at the top and actually matches up opposite complementary bases. And if we read each of these three bases at a time, we're able to see that now all I've got to do is go down and read the next three bases, then the next three bases, then the next three bases. And each time, it'll bring in an amino acid for me to build the protein. Pretty nice. So the specific steps are actually pretty simple. It begins when mRNA binds to the ribosome and we read our very first codon. So here's the bottom part of our ribosome. There's our mRNA, and our very first start codon, which happens to be AUG, is read. That forces a tRNA to come in with opposite bases, UAC, and it's carrying this amino acid, methionine. So that starts the process. We've started building our polypeptide. Step 2 is what's called elongation. We're going to elongate that chain and keep sticking amino acids on. So all we do is slide down. Notice this tRNA right here is empty. It's already given its amino acid and it leaves. Our next one comes in. There's UUU, which if we look up in our wheel, UUU codes for phenylalanine. So this tRNA brings it in. It locks this amino acid onto our chain. This is going to be our protein. Notice it already has 1, 2, 3, 4, 5, 6, 7 amino acids on there. Polypeptide means the same thing as protein for right now. Our ribosome is locking these guys together. And if you look, our next tRNA is coming in. There's GGC. If we go back and look up in our code what GGC codes for, It's a codon for glycine. So think of this guy as like a dump truck. It comes in, locks into place with opposite bases. GGC is attracted to CCG, the anticodon. That brings this guy in, this glycine, and it will be locked into place onto that polypeptide. And everything will just slide down, and we keep going again. Read the next three. Read the next three. And our chain keeps getting longer and longer. We're building our protein. So finally, that process is going to keep going until termination. When we get to the very end, or somewhere close to the end of our mRNA, we're going to reach a codon that is called a stop codon. It actually doesn't code for an amino acid at all. It codes for stop. So literally, at this stop codon, we'll notice if we look in our wheel, there's three of them, UAG, UAA, and UGA. A release factor, a special protein comes in which causes everything to be released. tRNAs leave, the protein leaves, and everything breaks apart. We're done. We have a long chain of amino acids. That's our protein. Maybe it's an enzyme, maybe it's a pigment, who knows what it might be. Structural proteins for muscles on the outside of a cell. It could be anything. But we followed the directions here and we've built it. Our cell followed directions from the DNA how to build a specific protein. Seems complicated, but it's pretty simple. As long as we can match up letters, we're good. So let's take a quick look at a video just to see this process in action. There's our ribosome. Looks like a weird Mario character. Our tRNA, which carries our amino acids, and this release factor, like we had said. So we'll initiate the process, we'll elongate it, like we said, and here's our termination. So here's our mRNA strand. We have that copy. Remember, it's a copy of our gene, our individual gene. We don't need to worry about this poly A tail, but at the end of them they have little protective caps. All that we will really care about for this purposes are these mRNA codons. Remember, they code for a specific amino acid in each one. So here's our small ribosome attaching to it. We begin reading our very first mRNA codon. There's our tRNA. Notice this little Mike and Ike looking thing. There's our amino acid. Here's our anticodon. That's going to be opposite, match up with the opposite bases of mRNA. That brings our tRNA in. Codes for met or methionine. It's following the directions in mRNA. Here comes our top half of the cap, our ribosome coming into place. Our builders are now ready to start building this protein. So all that we have to do now is read our next codon on our blueprints, bring in the proper materials. UUU, that's going to code for phenylalanine, or PHE. Notice the ribosome locks the methionine onto this guy, and the process keeps continuing. Lock it into place. We're growing. It's 3 amino acids. Now our peptide's 4 amino acids, and we keep sliding down, reading the directions. Pretty simple. So it's elongating, it's building, everything's looking good. Here comes our polypeptide, and that's going to, as we said, it's going to keep continuing. Our empty tRNAs leaving, new tRNAs coming in, and that process is going to continue until we get to our stop codon, which doesn't code for even for a tRNA, a release factor. UAG stops everything, we release. This gummy worm looking thing is our polypeptide, which is a protein that can become a specific shape. So in the end, what do we have? A polypeptide, a protein made up of amino acids that can be bent and folded into a proper protein shape. We're good. We follow directions. Bang.